All right. This is going to be our second Zoom recording on cellular reproduction. On your syllabus, I think this appears as cell cycle, or in bracket mitosis. Right. The basics of cellular reproduction. When you think of cellular reproduction, we're thinking of how organisms propagate their generations, or from like how a single cell organism will pass on its genes to its daughter cells. Multicellular organisms begin life as a single cell, and humans become trillions of cells because of cellular reproduction. And reproduction continues as we grow to replace worn out or damaged tissues. That means from the moment we are conceived to the moment we die, there is a higher chance that the cell that was existing at the time of conception might not be alive by the time we die, because every cell has to go through a lot of processes, and the cells have to regenerate themselves throughout our lives. Cellular reproduction. There are two forms of cellular reproduction. One is asexual reproduction and the other is sexual reproduction. A typical sexual reproduction is the fertilization of the egg by the sperm, that's sexual reproduction. When we say asexual reproduction, a single cell divides itself into two in a process called binary fission. But this doesn't require sperm or egg, and which is required in sexual reproduction. And as we have said in our previous Zoom recording, all cells come from pre-existing cells. And cellular reproduction is necessary for the production of both new cells and new organisms. Because from one single cell, the cells of the nervous system, the digestive system, or the respiratory system are created. From just one cell, different new cells are formed. And in some cases, new organisms. We give birth to babies because of this. Two important processes in cellular reproduction are growth and cell division. Growth is when the cell duplicates its contents, in particular its genetic material and the rest of the organelles. Whereas cell division is when parent cell contents divides into two daughter cells. And both processes are heavily regulated or highly regulated. You cannot perform cell growth and division without regular control mechanism. Cellular reproduction. At the heart of cellular reproduction are the chromosomes, or the genetic material. And when you say chromosomes, there should be DNA replication. That means the copying of the DNA material. Chromosomes, you need to double it and you will have 92 chromosomes. And then when the cell divides, each cell will have 46 chromosomes, which is the full set of chromosomes that was found in the parent cell. DNA is packaged into chromosomes, which is the complex of DNA and proteins, which allows easier distribution to daughter cells. As we said in our earlier Zoom recording, first we have the amino acids which form the codons, and those codons come together and form genes, and the genes form chromosomes, and the whole of chromosomes are called DNA. And DNA usually is wrapped around other proteins called histones, and that together forms the chromatin. And chromatin is all the DNA as well as the associated proteins. DNA is periodically wound around histones. These are the proteins I told you about. So DNA plus histones give us nucleosomes. It's like, let's say if you have a piece of stick, and if you wrap around something on it. Whatever you are wrapping around is the DNA, but that piece of stick is the histone. So the histone is like the skeleton that the DNA will be wrapped around. Just before cell division, chromatin condenses into chromosomes. We'll talk about this later. Humans have 46 chromosomes or 23 pairs of chromosomes. The cell cycle contains interphase, mitosis, and cytokinesis. This is an orderly sequence of stages from interphase, then to mitosis, and then to cytokinesis. And it takes place between the time a new cell has arisen to the point where it gives rise to daughter cells. Yes. At the time of interphase, there was only one cell. During mitosis, there will be a division of the genetic material, and then during cytokinesis, the cytoplasm will divide and you will have two daughter cells. So we have interphase and the mitotic phase. So this is the diagram. There's G1 phase, there's G0, there's synthesis phase, and there's the G2 growth, and then the mitosis. G1 is the first growth stage, whereby organelles are doubling. You double your organelles, your mitochondria, your Golgi apparatus, your endoplasmic reticulum. Everything will be doubled. Then if the cell is ready to go to the synthesis phase, when it reaches this phase, that means it has to replicate the DNA or it has to double the DNA. In G1, it was just doubling the organelles, or all the cellular machinery that's found within the cell will be doubled, except the DNA or the genetic material. I don't know. I wasn't sure if it was recording. So in G1 phase, everything is replicated. And then in S phase, the DNA is replicated or it is duplicated. But for some reason, if the cell decided not to go through this process, it will get into the G0 phase. G0 is zero growth, which means the cell is going to be arrested. It will not go through the cell cycle. It does not mean it's going to die. It means it's just going to perform its normal function, but it will not go through the cell cycle. During synthesis, the DNA is replicated. They will check if they have a proper number of chromosomes, and then it will go to G2. G2 is the point where you check if you have the right amount of resources to go through mitosis. When you go through mitosis, you have the PMAT, which is the prophase, metaphase, anaphase, telophase. It's an abbreviation. And then at the end, there will be the cytokinesis. So this pinkish part, it is called the interphase, including G1, S, and G2, that's interphase. And the mitosis is just this small part. And as you can see, the cell spends most of its time through interphases. Duplicated chromosomes are composed of sister chromatids joined at the centromere. This is like a belt that holds the two sister chromatids together. These are called sister chromatids at this stage. You had a single chromosome and there will be DNA replication. Replication is like a duplication. Now you have two and we call them sister chromatids because they are held together by a belt, centromere, and then during mitosis they divide, and now we call them daughter chromosomes. Interphase represents the majority of the cell cycle. It is the time when a cell performs its usual functions, and the amount of time varies widely depending on the cell. For example, if it is a cancer cell, it doesn't have much time to spend in interphase, so it goes through the cell cycle really quickly. And actually, it's like the cell cycle is not manageable. It is uncontrollably high. It goes through like G1, S, G2, mitosis again and again and again. Interface is divided into 3 stages, G1, S, and G2. G0 is only when the cells decide not to go through the cell cycle. Okay, G1 is the stage before DNA replication, and as I told you, in G1 phased, I keep saying phase, sorry. In G1, the cell doubles its organelles. It doubles its mitochondria, it doubles its endoplasmic reticulum, everything except the DNA. It accumulates materials for DNA synthesis, and then it makes a decision whether to divide or not to divide. If the cell decides not to divide, it will go to the G0 phase, which is the arrested phase, and it doesn't go on to division. But this does not mean the cell is going to go to apoptosis or programmed self days. Rather, it means the cell is going to perform its normal function without any cell division. Synthesis is the phase when the genetic material is duplicated or replicated. And this stage results in each chromosome being composed of 2 sister chromatids. And then the G2 is the stage following DNA synthesis, and it extends to onset of mitosis. And it synthesizes proteins needed for cell division. Yes. So G1, organelles are duplicated, synthesis, DNA is duplicated. G2, proteins that are relevant for mitosis or for cell division are replicated or synthesized. Okay. Cell division involves 2 kinds of divisions. The first one is mitosis, which is the division of the genetic material or the DNA, whereas cytokinesis is the division of cytoplasm. The mitotic phase or mitosis distributes duplicated nuclear contents of parent cell equally to daughter cells. What happens if the genetic material is not equally divided? Then we'll have a genetic abnormality. Each sister chromatid has the same genetic information, like the parent cell. And the daughter chromosomes are the previous sister chromatids. Once the sister chromatids are separated, we call them daughter cells or daughter chromosomes. Daughter nuclei. Daughter nuclei produced by mitosis are genetically identical to each other and to the parent nucleus because, let's say, some cell went through mitosis and it has 46 chromosomes like a human cell. I'm not saying the human cell went through our mitosis, I'm just saying, let's say it is one particular cell and it has 46 chromosomes. Now during the synthesis phase, these 46 chromosomes become 92 chromosomes. Then during anaphase, these 96 chromosomes are going to divide into 46. That means at the end of the telophase, every single, both daughter cells are going to have, or daughter nuclei are going to have 46 chromosomes. And these chromosomes are exactly the same to each other for the daughter nuclei, and they are also the same like the parent nucleus. Every animal has an even number of chromosomes because each animal gets those chromosomes from the parents, so they will have equal number of chromosomes. By the way, they could be odd pairs of chromosomes, but an actual number of chromosomes has to be even. For example, for human beings, we have 23 pairs of chromosomes. The pair can be odd, but the actual number of chromosomes has to be even, which is 46. Spindle. Spindles are part of the cytoskeleton we talked about in our previous Zoom recording. Spindle are part of the spindle or spindle fibers are made up of microtubules, and we talked about them earlier. Most eukaryotic cells rely on this structure to pull the chromatids apart during anaphase when you wanted to separate the chromosomes or sister chromatids. This is spindle fiber that the cell makes use of. We have 4 phases data, but in reality they are continuous processes. It's not like, oh, I finished prophase, now I need to go to metaphase, now I'm going to go. It's not like that. It's continuous, but it's not like clearly demarcated process. During prophase is the time when the nucleus starts to disintegrate and the nuclear membrane is gonna be like, it's gonna lose its normal intact structure. And when the nuclear membrane breaks down, that means the DNA lacks its protective layer, and the choice the chromosomes have is to condense themselves in pairs. Therefore, during prophase, there are 2 opposite scenes happening. One, the membrane is breaking down, the nucleus is breaking down. But because of this breakdown, the chromosomes don't have any choice rather than like sticking to themselves or to their pairs and then condensing themselves. That's why during this phase, we could actually see the chromosomes through microscope. That is usually not likely if it wasn't, the cell wasn't during prophase. The chromosomes might not condense because they have the protective layer, and within the nucleus, they can just be loose, they can be free. But once the protective layer is breaking down, they don't have any choice but to condense themselves so that they will be protected from exonucleases and lysosomes or other enzymes that break down the genetic information or DNA. During metaphase, one typical characteristic of metaphase is the chromosomes line up along the equatorial plate. It's like all the chromosomes will come like in the middle of the cell. And then during anaphase, these chromosomes, especially the sister chromatids, are pulled apart to the opposite poles of the cell, so that half of the genetic material will go to one side and the other will go to the opposite pole. And at last, during telophase and cytokinesis, during telophase, all the rest of it's like the nuclei, the 2 daughter nuclei start to be formed, and they appear 2 different cells instead of just one. And in fact, some structures start to appear, implying the cell is actually divided into 2 daughter cells. And when the cell reaches the cytokinesis phase, the cytoplasm or whatever is inside the cell membrane is going to divide into 2. This contains details of mitosis. I want you to go over them. I try to cover whatever is included over here when I was talking over here. So I want you to go through them because I'm sure I'm going to ask you a couple of questions from this part. So just to go over the notes here, the sentences, and check the diagrams. Phases of mitosis in animal cells. Although mitosis is divided into phases, it is a continuous process. I said that already. There is no clear demarcation between each phase. There is, but it's not always as clear as the diagrams try to show us. Always science tries to make models that makes the concepts easier, but in the reality is always more complex. DNA is replicated before mitosis begins. We said that already. DNA replication takes place during synthesis, the synthesis phase. Each chromosome consists of two sister chromatids attached at centromere. This is the belt that holds the two sister chromatids together. Red chromosomes, this one you don't need to know. Mitosis is usually followed by cytokinesis, and cytokinesis is the cyto part is the cytoplasm, and the kinesis part are moving apart. So the cytoplasm is moving apart, or there will be division of the cytoplasm. And it begins during telophase and continues after the daughter nuclei have formed. The daughter nuclei are formed during telophase, but the cytokinesis happens after telophase. Mitosis differs in plants and animals. It's obvious that plant and animal cells differ. We covered this concept in our previous Zoom recording. Plants have centrosomes, but they lack centrioles. This is a confusing part. This one, the centrosomes are the microtubule organizing center. Plants might not need the centrioles. These are like, we'll cover them in detail later, but these are parts of the microtubules that hold the chromosomes together and that move apart during the metaphase. For animals, they contain two centrioles that will move to the opposite pole. There are two centrioles actually in each pole, but during the prophase, that means two pairs of centrioles we have, therefore two centrioles will stay in one pole and the other two centrioles will move to the opposite pole, so that when the cell has to divide its genetic material, the microtubule that's attached to these centrioles will pull apart the sister chromatids. Cell membrane in plants forms from the center outward along a cell plate. Here is the thing I want you to remember. Cytokinesis in plants happens through the formation of cell plate, whereas in animals, cytokinesis happens through cleavage furrow. For animals, it happens through cleavage furrow, and for plants, it happens through cell plate. This is cytokinesis. Cytokinesis happens this way. Yeah, this is detailed information about metaphase, anaphase, telophase. Cytokinesis in animal and plant cells. Cytokinesis accompanies mitosis in most, but not all cells. There are cells that go through mitosis, but not cytokinesis. That means if you go through mitosis, that means you divided your DNA into two and you are going to have two nuclei. But if you didn't go through cytokinesis, that means the cytoplasm of the cell is going to remain intact. That means there is a single cell, but you have two nuclei, and that makes that particular cell a multinucleated cell. So there is a mistake on your slide because mitosis with cytokinesis doesn't result in a multinucleated cell. A multinucleated cell says A, that means it is one cell and it has a lot of nuclei. That happens only when there is mitosis, but there is no cytokinesis. So mitosis without cytokinesis results in a multinucleated cell. I want you to remember this. Examples of multinucleated cell is a muscle cell in vertebrate animals, or embryo sac in flowering plants, which is a multinucleated cell. Cleavage furrow. The easiest way to remember cleavage flow in animals is to just have a balloon full of water and then to squeeze it in the middle so that it appears you have two different balloons. That contractile ring is formed because of the cleavage flow, and this is more common in animals. We don't see it in plants. In plant cells, cytokinesis happens through cell plate formation. I don't know if you could see, there is a line in the middle of this particular cell. This line, cell plate, is gonna grow larger and larger, and it's gonna be both the cell membrane, and later it will develop its own cell wall. So this is how the cytokinesis in plants happens. First, there's a cell plate formation, and then it's going to grow into plasma membrane, and then it's going to have its own cell wall. Plant cells have rigid cell wall that prevents furrowing, which is more common in animal cells. It involves building of new plasma membrane and cell walls between daughter cells. Said that already. Golgi apparatus produces vesicles. We covered this from our previous slide. And cell plate is the newly formed plasma membrane, and later it helps in the formation of the cell wall. New membrane releases molecules that form a new plant cell walls. Yeah, we said that already. The cell cycle control system. The cell cycle must be controlled. It is a heavily regulated process. You cannot perform cell division, or you cannot go through cell cycle without any proper control system. This ensures that the stages occur in order and that the cell cycle continues only when the previous stage is successfully completed. That's why we needed checkpoints. If G1 phase is not going well, you cannot go to synthesis phase. If synthesis phase is not right, you cannot go to G2 phase, or you cannot go to mitosis phase. So what are the cell cycle checkpoints? We have three of the many cell cycle checkpoints we need to know. One is the G1 checkpoint, second G2 checkpoint, and then the mitotic stage checkpoint. G1 checkpoint, if you finish G1 checkpoint, that means you are committed to divide after this point. If the G1 checkpoint passes, that means the cell has decided to move on the cell cycle. If the checkpoint is not passed, or if it fails the checkpoint, that means the cell is going to enter into the zero, or the G0, the zero growth stage. Proper growth signals must be present to pass, yes. If there are no proper growth signals, like if the cell doesn't have the proper organelles or the right amount of nutrients, then the cell is not going to go through that process. And DNA integrity has to be checked at this stage. If the repair is not possible, there will be apoptosis, which is a programmed cell death. G2 checkpoint. So if the cell checks out for G1 checkpoint, then we have to check for G2 checkpoint. This verifies that the DNA is properly replicated. If there is any DNA damage, it has to be repaired. Then if the cell passes all these tests, it is going to go to the mitotic stage, because from G1 to G2 there is synthesis. If G1 is passed, it will go to synthesis, and then if G2 is passed, it will go to the mitotic stage. Mitotic stage checkpoint is found between the metaphase and anaphase, and all chromosomes must be attached to the spindle to pass. Okay. One defining feature of the mitotic stage checkpoint is that all chromosomes have to be attached to the spindle fibers at the centromere. If there is a chromosome that is not attached to a spindle fiber, then this mitotic stage checkpoint might tell the cell to stop going through the cell cycle, because there will be a genetic abnormality if there is a chromosome that's not attached to spindle. Okay, this we have seen already. This is the G1 checkpoint. If it passes, it will go to synthesis, and then there's G2 checkpoint. If it passes, it will go to mitosis, and there's mitotic checkpoint. If it passes, it will go to anaphase. If not, the mitosis stage will be aborted. Okay. Internal and external signals. The cell cycle has, as I said earlier, it has a heavily regulated control system. And this control system has internal and external signals. And a signal is a molecule that stimulates or inhibits an event. The signal could be pro or against a certain event. External signals, they come from the outside environment. Internal signals come from inside the cell. One way of making signals is to add a phosphate group to a certain molecule. So when you add a phosphate group to a certain molecule, it makes that particular molecule to be known by other control or other regulatory proteins. So kinases, they remove a phosphate from ATP and they add it to another molecule. You might need to know this for your molecular biology class in the future. Kinases, they add or remove a phosphate group. Cyclins, as the name implies, if you have a cyclin, that means that cell is going to go through a lot of cell division again and again. These are internal signals present only during certain stages of the cell cycle. So if you have cyclins, you are going to go through the cell cycle indefinitely. That's why they are present only when they are needed, or only at certain stages of the cell cycle. So for this reason, cyclins has to be destructed, or that we have to destroy the cyclins. Like after we use them, we have to destroy them. Otherwise, the cell is going to go through an unnecessary cell cycle. External signals. One external signal is epidermal growth factor, which stimulates the skin near an injury to finish cell cycle and repair injury. The hormone estrogen is also an external signal because it's produced outside of the uterus, but it will be transported to the uterus so that the uterus could prepare itself for egg implantation. Apoptosis, I said that earlier, it is a programmed cell death. A cell cannot live forever unless it is a cancer cell, and even a cancer cell cannot live forever because at some point there will be scarcity of nutrients and resources, and it will die at some point. But every living cell has a time limit, so there is a programmed cell death. And once a cell destroys itself, the remaining cell fragments are engulfed by white blood cells like neutrophils, basophils, and things like that. Apoptosis is unleashed by internal and external signals. We said that already. For example, if you are exposed to nuclear radiation, the cell might kill itself because it is an external signal, and if the cell doesn't destroy itself, it might send wrong signals to other cells, which might kill the whole organism. Internal signals could be one simple example, could be a hormone. A hormone could serve as a signal that will go to certain cells and tell them to destroy themselves. Apoptosis helps to keep the number of cells at appropriate level. If there is no programmed cell death, that means cells are giving rise to a lot of daughter cells and still the parent cells are living. That means we are going to have an unusually high number of cells, which might lead to a lot of abnormalities, and at the end of the day, the days of the organism. They are normal part of growth and development. Apoptosis is normal. Every cell goes through some kind of programmed days at some point. This is apoptosis. Cell cycle is regulated by signals that inhibit or promote cell cycle. Yes, cell cycle, as we have said earlier, it is a heavily regulated process to support it or against it. Cancer may result from imbalance. Imbalance in essence, cancer might occur because we don't have signals that inhibit cell cycle, or we have signals that promote cell cycle. It's like the absence of certain signals might cause cancer, and in some cases, the presence of some signals might cause cancer. Cancer is a disease of the cell cycle in which cellular reproduction occurs repeatedly without end. Here is how a cancer cell develops. There is one mutation, just a single mutation, and then that mutation leads to 2 mutations, and then 3 mutations, and then those mutations are converted into an invasive tumor that could go to other tissues. It could be malignant tumor, and then the malignant tumor is really dangerous tumor, and it could go to other parts of our body, and it becomes a distant tumor. And these distant tumors, we call them metastatic tumors, or tumors that has gone to other parts of the body. That means they are not localized to the certain region they first start. Proto-oncogenes and tumor suppressor genes. Okay, I'm gonna give you an example that is really, it is related, but it's not related to biology. When you say crime, a crime is failure to do what you are supposed to do, or failure not to do. Okay. Let me see if there's an easier way to say it. Okay. A crime is there is something that the government wants you to do, and if you fail to do it, that's a crime. And another side of it is a crime is there is something that the government don't want you to do, but you do it. Either way, it is a crime. So this is the proto-oncogenes, and tumor suppressor genes are the same. The proto-oncogenes are the ones that do things that they are not supposed to do. Pro-to-oncogenes is like the way I see this is pro-to-on. Pro is it supports, to on, not off. That means it supports the growth of the cell cycle, pro to on. These oncogenes, they code for proteins that promote the cell cycle and inhibit apoptosis. It's like they are telling the cell to do to go through the cell cycle again and again. And when the cell tries to kill itself, it's like telling her, no, don't do that, just go on. They are often likened to the gas pedal of a car because they accelerate the cell cycle. So this one, they do what the government don't want them to do. Tumor suppressor genes, these are when the police are not doing their job. These are supposed to code for proteins that inhibit the cell cycle and promote apoptosis. That means they want the cell not to go through the cell cycle, and they promote apoptosis if this is necessary, but these genes, they are not doing their job. They are supposed to suppress the cell cycle and promote apoptosis, but they are not doing their job. Either way, we're going to have cancer because of that. When proto-oncogenes mutate, they become cancer-causing genes called oncogenes. When a tumor suppressor genes mutate, their products no longer inhibit the cell cycle or promote apoptosis. Okay. This is how the proto-oncogenes and tumor suppressor genes in the cell cycle work. Other genetic changes in the cancer. In every chromosome, not every chromosome, some chromosomes have endpoints called telomeres. These telomeres are, let's say, if you have a chromosome, this is one chromosome, and they have telomeres at these ends, at the ends of the chromosomes, both ends. And every time a chromosome goes through replication or duplication, these telomeres get shorter and shorter. And that's normal. Every time a cell goes through DNA replication, these telomeres get shorter. But in some cases, even though you go through duplication, these telomeres don't get short. So when there is absence of telomere shortening, that means this chromosome is becoming cancerous and the whole cell is becoming a cancer cell. So absence of telomere shortening could be the source of some cancer cells. Chromosomal rearrangements. Chromosomal rearrangement this is easier to explain it through radiation. If you have one atom and within that atom there are neutrons and protons, but if it is a heavy atom with a lot of protons and neutrons in it, the nucleus is unable to carry those particles and it becomes unstable and it's shaky. And in the process, the nucleus might lose some of the neutrons and protons. And these protons and neutrons might reattach to another chromosome, or in this case, to another element, and they might form an abnormal chromosome. So when chromosomes of cancer cells become unstable, like the unstable nucleus, portions of the DNA double helix might be lost, duplicated, or scrambled. And then when a portion of this chromosome may break up and reattach to another chromosome, these events are called translocation. Just a portion of one DNA helix is breaking away and joining another chromosome within the helix. These are cell cycle genes that are associated with cancer. BRCA1, BRCA2, you don't need to know this. Cancer cells, the development of cancer is called carcinogenesis, and cancer cells lack differentiation. In fact, they don't have any function. Their only function is to divide themselves uncontrollably, and this makes the normal functioning of the cell really difficult. They might be mortal, not like literally mortal, but compared to the other cells, they look like immortal. And they have abnormal nuclei with abnormal number of chromosomes. Cancer cells don't undergo apoptosis. They don't go through programmed cell death, and they form tumors, and they do not respond to inhibitory signals. That means to have signals that try to inhibit the formation of tumor, the cancer cell ignores these signals. And they undergo metastasis. Metastasis is when one cancer cell breaks away and migrates to another tissue. It's called metastasis. And then it forms new blood vessels. Angio is, by the way, a blood vessel. Genesis is formation, so formation of new blood vessels. It's like there is a tumor right here. It breaks away, goes to a different tissue, it clears the forests and establishes its own transportation system, and then it nourishes itself. That's metastasis. They travel to start new tumors, and they form new blood vessels to nourish themselves. These cancer cells might be benign or malignant. If it's benign, that means it is within a capsule. And even if they are dividing, at some point they stop dividing because there is a capsule that holds the tumor. But if it is malignant, it breaks this capsule and it will go away to different tissues, it will go away from the cell, and it will metastasize. That means it will form its own blood vessels and nourishes itself, and it will form any tumor. This is the development of breast cancer, which is benign tumor and become malignant tumor and then breaks away. And yeah, this is metastatic tumor. It was small and then it's growing, becomes malignant or bad, and then breaks away, like you can see from picture. Can we treat cancer? Obviously, we can treat cancer by removing the tumor if it didn't metastasize already, or we can interfere with the ability of cancer cells to reproduce. One thing you need to know is cancer cells are not strong compared to regular cells because they didn't finish the regular cells development. They actually go to division before they finish their actual growth. The biggest problem is the uncontrollable cell division. You can actually kill them easily, but the division is uncontrollable. And when you kill one, you have like millions that are that has metastasized or that duplicated themselves. They're rapidly dividing cells, and they're susceptible to radiation and therapy and chemotherapy. We can damage their DNA and some aspect of their mitosis, but doing so might lead to certain side effects for the patient. And these days, where there's a development of hormone therapy designed to prevent cells from receiving signals for continued growth and division, because after all, cancer cells are also cells, and any cell cycle requires hormonal regulation. So if you develop a hormone that could interfere in the cell cycle, that could be beneficial for the organism, and we could be able to disrupt the growth and division of the cancer cell. What are the protective behaviors and protective diet in cancer prevention? Avoiding smoking, avoid sun exposure. If you are a heavy drinker, you should stop that. Protective diet, doing weight loss exercises, having foods that have a lot of vitamin A and vitamin C, avoiding processed foods, and then the cabbage family are also found to be protective of cancer cells. Okay, this is all about the cell cycle. What I want you to remember, as I have said in the earlier Zoom recording, is just focus on the big picture. I'm not going to ask you a detailed question because this is just a lab part, and the quiz, I'm not going to ask you every single detail. Just focus on the big picture. That's all I have. Yep. Thank you for listening.